Good morning. I'm Daryl Brugink, Executive Editor with No-Till Farmer, and I'd like to welcome you to today's webinar, where we will be learning about the impact of biological seed treatments on plant development and yield. Now, farmers in general use seed treatments a great deal for disease, insect, or nematode control, but perhaps not as up to speed on what a biological seed treatment might be able to help them accomplish in a no-till system. Today's No-Till Farmer webinar is sponsored by Midwest Biotech. And here to help us learn more about biological seed treatments is Doug Miller, Vice President of Midwest Biotech. Doug has done a number of webinars on varying topics with us, and he's done some teaching in his past as well. So he always does a great job of presenting these webinars. We look forward to hearing from Doug in just a moment. Thank you again for joining us, and I will turn over the controls to Doug. Good morning to you, Doug, and thanks for being here today, right here in our offices at No-Till Farmer.
Thanks, Daryl. Always a pleasure to be here with the folks at Lessiter and lovely downtown Brookfield, and such a nice warm day. But we're glad to be inside and have a chance to talk about soil biology and plant health, and in particular about biological seed treatments. A number of things we want to talk about just to set the story up. We'll talk a little bit about the steps involved in seed germination and early plant development and root growth. Most farmers are going to know a lot about this anyway, so we're not going to spend a lot of time, but there are a couple points I wanted to make. And then more importantly, we want to talk about those biological factors that affect root activity and plant growth, the things that are going on in the soil. A lot of attention lately on cover crops and root exudates and the benefit of having living roots growing in the soil more of the time throughout the season. And we'll talk a little bit about that, but that's been pretty well covered by the no-till folks through the Conservation Tillage Guide and the newsletter. And if you're still a little bit unfamiliar, I'm sure Daryl's got a few books he could sell you on the topic. But beyond that, I want to spend more time talking about some of the other things that are associated with root growth and biological activity in the soil that do affect plant health and ultimately yields. So we'll focus on some of this that's actually pretty new research. A lot of it's just been reported in the last 2 or 3 years, and so it helps explain some of the things we've seen over the past 35 years since we started working with biological seed treatments and some of the other biological tools that we started out on our own farm and have been actively marketing through Midwest Biotech since the early 80s. But I did want to get into a discussion of the different types of biological seed treatments. That it's become almost a catch-all category. Lots of people will advertise that they've got a biological seed treatment, but there are many different modes and many different ways in which they're working on the seed to affect crop growth and root development and ultimately yield. And so I just wanted to take a little time to discuss the different types, the pros and the cons of the different alternatives, and break down this pretty large category into sort of a more manageable subset of groups, so we can talk about what they're going to do for you. And then finally, we'll spend a little bit of time talking about our seed treatment. We don't want to make this a 50-minute sales job, but we will talk a little bit about our experience in seed treatments, and that's been through the Chandler product that we market. But we'll leave that to the end. And as well, a number of the people that registered had typed in questions already. We encourage, if you do have questions, to go ahead and we'll try to get to those at the end. And for those who provided questions in advance, we'll try to touch on as many of those as I present, so we can get those responses out to you while we're going. But anyway, we'll launch into it. And just as a reminder, we'll talk a little bit about seed germination. Basically, there are 3 requirements once you put the seed in the ground. It's dormant tissue. Abscisic acid or some other biochemicals are keeping that seed from germinating until it reaches the required situation. Basically, you're going to need oxygen. You're going to need some warmth. The range of temperatures at which seed will germinate depends on the particular plant type. There's usually an optimal rate at which the seed's going to germinate as quickly as possible and get that plant started soon, but the temperature can vary quite a bit. Water is a little bit more critical, and the amount of water required depends on the particular type of plant. For example, corn has to get to about 30% moisture content before it's going to germinate. If it doesn't reach that level, then if it gets started but doesn't reach 30%, then you're going to have kernels that are out in the field ungerminated and eventually might rot and won't do you any good. So there has to be that moisture available. Soybeans actually require a little bit more, and wheat even more. And kind of what drives the amount of moisture is the plant material within the seed. Corn starch based, soybeans are lipid based, and wheat has a higher protein content, and so it needs more moisture to germinate. All of these three things together point to the importance of seed soil contact and getting that seed bed, the seed slot, I should say, opened up, provide ample space, but you don't want to get it too tight or you won't have oxygen. You do have to have good content or good contact with the soil, because basically the seeds are going to draw that moisture from within about 4/10 of an inch of the seed. They're not going to be able to reach out very far to get that moisture, so it has to be close. With our product, it's talc based, our dry products, so the talc will help attract moisture. Other seed companies are using gels and other coatings to help attract moisture. And in the past few years, it's been dry, so this has become kind of an issue. But beyond those basic requirements, once you meet the minimum of moisture and temperature, germination is triggered by a plant hormone called gibberellin. And basically, the gibberellin tells the enzymes that they've got to get busy, break down the seed coat, start to digest the starch and the other energy in the seed. And that's going to be the main food source for this plant as it germinates and starts to emerge, until the root system's large enough to start providing nutrients and other resources through the roots, and then won't have to rely on that seed source. So that's one of the reasons why we like to develop a root system early and get the plant out of the ground soon, because there's only a limited amount of energy for plant growth available in the seed itself. Once those enzymes start to activate and break that material down, it's only going to be available for a short period of time. And so you want to start drawing nutrients into the plant from soil sources by getting that root system off to a good early start. One of the things we'll talk about a little bit later is the importance of micronutrients in all of this. Basically, we need micronutrients as composition for some of the biochemicals and the cell walls and some of the structures in the plants, but micronutrients also activate the enzymes. Basically, the enzymes are dormant until they're activated and start to work, and typically most enzymes require a specific type of micronutrient, whether it's magnesium or manganese or boron. Copper is very important for root growth, so copper is typically one of the activators for enzymes used in root development. So that's one of the key roles for the micronutrients, and there's been a lot of attention paid to that in crop farming in the last few years. How do we make sure we got enough micronutrients available? How do we apply them? And so that part of their key role is really in activating these enzymes that trigger all of the different biochemical processes that get that plant up out of the ground and start sinking roots deeper into the soil. As far as early root development, everybody should be pretty familiar with the role of the initial radicle emergence. That's one way of defining germination, is once the radicle emerges from the seed, then it's germinated. Interestingly, you can get academics to debate anything, and there are agronomists and plant scientists who have different definitions of germination. But for the most part, once the radicle emerges, you're going to start to develop that root system. In corn, a few days later, the primary or taproot is going to form. Seminal roots are going to emerge from the seed layer and start to sink down, and those things are going to be the foundations for the root system that develops and starts to feed the plant. little bit later, crown roots are going to form on the stem above the seed. And then by the time the plants mature, typically you've got a good 2 or 3 layers of crown roots. Crown root development is important not only for providing nutrients to the plant and also for the interaction with the biology, but lateral crown root development is going to help stabilize that plant in addition to the brace roots that emerge much later, typically about 6 weeks after emergence in corn plants. So there's a number of different stages of root development that occur early. What we'd really like to talk a little bit more, other than the physical stages, are those biological aspects. The plant root system does provide structural stability, so those crown roots that move out away from the plant laterally are really more important for keeping that plant upright and stable in the presence of wind and other weather, probably more so than the brace roots. And of course, the hair roots provide structure, the sort of the traction for the roots as they grow through the soil levels. As we know, based on the discussions about the impact of cover crops on loosening up the soil and improving soil structure, roots can exert a great deal of pressure. In fact, most roots will exert about 100 pounds per square inch at the tip and can exert a much more. In fact, you can have a compacted soil that, as long as it's got less than 300 pounds per square inch of soil pressure, the roots can still get through it. Not all the roots, but some. So the roots have a great deal of strength, and basically the hair roots, in addition to providing source of nutrient and water, provide the traction so that root can progress through the soil. The tap or primary roots are going to provide a lot of the water sources from especially subsoil areas. So if you run into a dry spell like we had last August here in the Midwest, if you've got an adequately deep taproot system or primary root system on the plant, you're going to be able to draw moisture even though the surface sources have dried up, and you keep that plant going, keep it from going dormant, and keep it producing until you can get to a time when you've got better weather and more stable moisture. That's sort of the established knowledge on root systems. And again, we're not going to spend a lot of time on it. We're going to go fairly quickly. But more importantly, we want to talk about the soil biology and soil health through the rhizosphere. That's the area right around the root system. A lot of people define it as being within just a couple of millimeters, or basically the thickness of about 2 dimes from the root surface under the plant. The more roots you have, the larger the root system, the more potential you have for interaction between those soil microbes and the plant. Those folks involved in the cover crop business and through no-till farming have focused especially on the importance of root exudates. Root exudates are basically carbon, in some cases they're polysaccharides emitted by the plant, and that's basically a food source for the soil microbes. Root exudates also include dead cells that are sloughed off from the roots. It's also the root cap, the very tip that's pushing its way through the soil, has to be updated and regrown. So those old root cap cells are going to serve as a source of food for the microbes. And there's also mucilage. It's sort of a sticky, gluey-like substance that surrounds the root and provides a little bit of lubrication as it pushes through the soil. And the scientists tell us that even after the cells are sloughed off and released from the root and new ones are grown, that those old cells are still generating mucilage, and that's a carbon source for the microbes. So all of those different exudate sources are going to feed the microbes, and in turn the microbes, through their activity for immediate purposes, convert nutrients, make them available to the plant, and then exchange those at the root surface at the rhizosphere. So that's how the nutrients enter the plant, is they're basically through the microbes bringing them to the roots, and then they go up through the xylem and into the plant structure. Again, I'm not going to spend a great deal of time on this because the folks at No Till Farmer have spent have covered it very well in the conferences and the publications. And so that part of the soil biology, I think, has received a lot of attention, and we don't want to pay just recover familiar ground. Some of the additional biology, especially a lot of new research results, um, that's what I want to focus on in the next couple of slides. There are other processes going on that are related to the root system that affect not only soil health, but plant health and ultimately yields. And so we want to spend a little bit more time on that. In particular, one of the things that happens in the rhizosphere, that area right around the root system, is chemical signaling. The plant and the microbes are signaling back and forth using different biochemicals. In the soil, there are probably, I've seen estimates of about 10,000 different species of beneficial microbes. If you include all the subspecies and different types, I've seen estimates as much as about 800,000 distinct types of microbes in the soil. So, it's a huge population. In terms of their numbers, the rough estimate is that if you take up a hand of soil, you're going to have about as many microbes in that handful of soil as there are people on the earth. Numbers in the billions. So there are lots and lots of guys active in the soil. The vast majority of those are beneficial. They're going to work to the benefit of the soil and the plant. The minimum, or I should say the minority, are pathogenic or harmful. They cause plant disease and other problems. Basically, the good guys keep the bad guys in check. So the more healthy your soil is, the more beneficial microbial activity you have, the less trouble you'll have with the bad guys. How they interact is pretty interesting, and it's through these chemical signaling processes that the plant decides what it needs and then sends out a signal to those microbes specifically suited to handle that task. So through the exudates from the roots, the plant signals which particular microbes are going to be allowed to come into the rhizosphere. So in the full soil profile, tens of thousands of different species, but only 1,000 or so are actually invited in to be close neighbors and work with the plant at any given time. And that group that's invited into the rhizosphere is going to change. It's going to depend on soil type. It's going to depend on the type of the plant. It's going to depend on the plant's specific needs. So the plant scientists tell us that if they take two plants from the same type of soil, but they're different plant species, they're going to find different microbes around the roots. You can take same plant, different soil type, and you're going to get different populations. So the chemical signaling helps determine who's invited in, and the chemical signals are also going to repel those other microbes that aren't needed and keep them away, so the required helpers can get in there. So if the plant needs micronutrients, sends out a signal, and it's going to try to get that nutrition from the microbes that are going to help pass it along into the root system and up into the plant structure. If there are toxins present, there was recent research about 3 years ago, I think it was out of Germany, that showed that if there are toxins in the soil near the roots and the plant's going to have to deal with that, it's going to signal particular bacteria species that can eat up and handle those toxins and get rid of them, so the plant isn't going to be affected by it. So the signaling can be triggered by all sorts of different stimuli, needs by the plants, either for nutrition and promotion of growth, and as well to try to handle different threats that are coming its way. So again, all of this highlights the importance of soil health. The more microbes you have growing in the soil, the better your soil health is, the better the plant's going to be able to deal with its challenges and promote plant health. There are actually a limited number of microbes that are actually allowed to colonize within the roots. The two most common things we might think about are, first, the rhizobacteria that form nodules on soybean roots and other legumes to fix nitrogen. That's pretty well known. They're signaled in by a chemical known as flavonoids. The plant produces flavonoids, and it says, hey, I need some rhizobacteria. It's time to fix nitrogen. Once the plant knows that the rhizobacteria are in the area because of signaling back from the bacteria to the roots, the plant opens up fissures or gaps in the root system that allows the bacteria in, and then the colonies form. The more that the plant's able to signal back and forth, and the more rhizobacteria that are present, the more nodulation that's going to occur. The other one that gets a lot of attention are the mycorrhizal hyphae, the fungal species that shoot out their long roots, the hyphae that extend actually several feet in the soil and not only provide nutrients to the plant, and the hyphae can extend within the cell gaps in the outer wall of the roots, but there's also evidence that they're useful in signaling other sorts of things. We reported a study that was published just here in the past year by some folks at the University of Aberdeen who looked at plant defense chemicals that are emitted by any plant, and it's used to help protect the plant from insects or other predators that might harm the plant. And in particular, I'll just go back for a moment. The hyphae extend throughout the soil, and again, they can be several feet long. This particular study, the purpose of it was to find out how plants are triggered to defend themselves if a pest comes into their environment. And it's known that if an insect shows up or a disease organism shows up, the plant starts to send out different chemicals that will either deal with the threat or at least try to repel it. And Dr. Johnson and the other folks at the University of Aberdeen first thought that it was a signal sent through the air, some sort of chemical emitted by the leaves. Because what they noticed was that if a plant was infested, its neighbors also started to produce these protective chemicals, this defense mechanism. So if your neighbor's infested, you get ready to deal with the threat as well. And they first thought that the signals between the plants were emitted by chemicals that were picked up through the air. So they bagged the plants, they used very fine mesh bags, made sure that the plants weren't able to exchange any sort of gaseous compounds that would be a messaging system. And if then they infested one of the plants with a pest, an insect or something like that, and found out that the neighbors were still communicating and producing the defense chemicals. Well, then it must be going through the soil. So it's either by root contact or something like that. So they put the roots of the plants in fine mesh baskets and then varied the fineness of the mesh. And as they got to smaller and smaller and smaller mesh, the plants were still able to signal. The roots weren't able to penetrate through the mesh, so the roots were not in contact. The mesh got so small that bacteria couldn't be causing it. And the only organisms small enough to go through the mesh at the very end were the mycorrhizal hyphae. And so effectively, the fungus were forming an underground network that allowed one plant to communicate with another one when it faced a threat. And so that again highlights the importance of soil health. Anything you can do to promote not only bacterial species, beneficial bacteria, but also the beneficial fungus, it's going to help with plant health because the plants evidently can communicate that they're facing a threat, and they've got to get geared up to try to repel it. This is very new research. Again, it was just reported very recently in Ecology Letters, and the popular press picked up on it. I saw it in The Economist magazine, which is a European publication. And there's a lot to be learned, but it still does emphasize that soil health is pretty important for a number of different reasons. One of the things we talked about in September in our webinar was the fact that fungal species are very sensitive to tillage, and the bacteria recover more quickly than the fungal species. If anytime you put steel in the soil and you start breaking up hyphae and disrupting those fungal populations, they will rebound, but it takes a while. And so anything you can do to promote the health of those organisms, including no-till farming, cover crops, and of course, we'd like to think that biological products like ours fit in. Any of those activities are going to help improve not only soil health but plant health, and that has to be good for the bottom line when you look at yield. Other sorts of chemical signaling that occurs within the plant, some of which comes through the plant hormones. We talked about gibberellins and their role in germination. The two other main categories of plant hormones are auxins and cytokinins. Auxins are generated within the plant and are the key hormones for signaling root growth. They control growth of the root system, so anything you can do to promote auxins within the plant is going to be beneficial for root growth. Cytokinins are really associated with growth above the ground, shoot development and growth. The hormones get a little bit complex because it depends not only on their presence but on their ratio. Cytokinins and auxins work together, but the mix of the two determines what's going on in the plant. If the ratio, if you've got relatively few cytokinins to auxins, you're going to get cell elongation. So the plant's growing, but it's not generating new tissues or new cells. If the cytokinins increase, which happens at particularly critical phases as the plant starts rapid vegetative growth, or at the end of the season when it's shifting from vegetative to reproductive growth, that's when a lot of cell division occurs. And so the cytokinins have to be high relative to the auxins. The complexity is even deeper. There's recent research reported in the last few years that shows by itself cytokinins actually inhibit root growth. And so you say, well, if how can we grow big root systems if we got cytokinins running through the plant? Well, that's just a one part of the equation. The cytokinins by themselves may inhibit root growth, but you've also got the auxins and all of the biochemicals working. So in isolation, cytokinins deter root growth, but there are lots of other things going on. So it's a very complex relationship, not only among the hormones in the plant. And again, recent research in the agronomy literature has shown that cytokinins not only are produced in the plant, they're actually generated in the root tips, and then they travel up the xylem into those areas where you've got actively growing shoots. So they're generated in the roots. They inhibit root growth by themselves, but they're produced there. And as well, synthetic cytokinins are produced, as it seems now, by the microbes and then transmitted into the roots just like they transmit other nutrients, and then those can flow up the xylem and further promote plant growth. So again, soil health not only affects nutrient absorption into the root system, but it seems to affect the hormones that are used for growth as well. So a healthier soil, more microbial activity should promote root growth in a couple of different ways. So again, we're not the sort of thing you typically hear about because a lot of this research is relatively new, but we're finding new ways in which soil health and microbial activity affects the plant and ultimately the yield. Some of the particular chemicals that are used for defensive purposes by the plant work in different ways. Flavonoids are those chemical signals that attract the rhizobacteria toward the roots, and for legume type crops signal that, hey, the plant's ready to be colonized and nodules are formed. So the flavonoids are related to what are called nod factors that tells the plant that, hey, it's time to form nodules. But flavonoids also inhibit disease, and so they repel certain types of fusarium and some of the sources of soil-borne diseases. So they serve both a beneficial role by sending positive signals to the good bacteria, but they also work to repel or slow down some of the bad guys. Tannins, most people encounter tannins if you bite into a grape seed or you taste sort of unripened fruit. There's often a very very bitter taste. It puckers your mouth. Tannins are used as a natural preservative in lots of different things. And basically, it's a defensive mechanism. Not only does it taste bad to the insects, but more importantly, it blocks key digestive enzymes in the insects, so that as they consume that fruit, or they consume those parts of the plants that contain tannins, the insects aren't able to get any sort of energy out of it, and so they're going to have to move on. So the presence of tannins deters insects, and it's also been found that it's got something to do with deterring microbial populations, the pathogenic or the bad ones. Um, Diterpenoids are another form of biochemical that plants will produce that have antifungal and antibacterial properties. So if the bad guys come along, the plant will fire up the diterpenoids and try to keep the bad guys away. And as well, the plant can produce particular proteins and enzymes that block other proteins and enzymes that are key for the insects or the microbial populations that are pathogenic and trying to harm the plant. So there are many different ways that the plant can defend itself naturally by using these different biochemicals. And we're just really starting to learn about a lot of this. And there are a few commercial products available that are based on applying these sorts of chemicals. Mode of delivery, when to do it, timing, all of that sort of thing is relatively new, but again, it provides another way to try to protect plants and keep them healthy, so that you can improve yields and have a more profitable situation. In terms of the different seed treats that are on the market, like I say, everybody's sort of using this catch-all category, biological seed treatments, and they're all generally intended to improve plant health, but the modes of action are different. And just to sort of simplify things and talk about some of the differences, what I'd like to do is break this down into four categories. The columns, control and promote, indicate whether the particular treatment is trying to control the pathogenic or the harmful microbes, or whether they're trying to promote the beneficial ones. We could label these um, probiotic and antibiotic, but those terms are used in the medical industry, and I think especially antibiotic has some bad connotations. So I don't think in the ag business we really want to get into using those terms, but it's really the same idea. I'm either controlling bad or pathogenic, harmful microbes, or I'm trying to promote the beneficial ones. And then the mode of delivery is either through a chemical or a biochemical source, or by using live cultures of actual microbes. And so if we start in the upper left corner and talk about the biochemical controls, that's probably the most familiar class to us. Those are the fungicides and the insecticides people have used for years. There are lots of new products in the market. We know they work because of their longevity on the market. Most of the newer products are intended to try to target in on specific problems. Sort of the older class, especially of fungicides, had multiple modes of action. The problem was that you typically were harming the beneficial fungus and the beneficial microbes while you were also taking out the bad guys. And a lot of the newer ones have a single mode of action that targets in on a specific enzyme or a specific biochemical in the insect, or in the fungal species, the pathogenic fungus, the bad guys. And by working very narrowly, those probably have less impact, or it's believed they have less impact on the beneficial populations than the sort of the older um, technology. Now, the problem or the downside with that is, since you're just working on one very narrow mode of action, the pathogenic fungus or the insects can alter their genetics and become resistant to those particular chemicals. So that's the problem we're facing, is through those biochemical controls. They work great. They probably don't harm the beneficial guys quite as much, but their mode of action may not be too long lived. You may have to switch to other products, or hopefully they'll come along with different tools later on, because resistance can develop fairly quickly. Another fairly new category are live culture biofungicides. One sort of product where you actually apply a live culture of bacteria or fungus that either competes with or actually feeds on the harmful guys. So it's sort of like the idea of putting out wasps or other beneficial insect species on the surface to feed on aphids or other harmful insects. So the same thing can go on below the surface in the soil, where you actually use a live culture of a beneficial bacteria or fungus to drive away and or at least compete for resources with the bad guys and starve them out. That's a relatively new class. Um, on the beneficial or promotion side, people are probably much more familiar with bacterial or fungal inoculants. That's where we actually put on live bacteria on the seed, and typically it's in legume crops where you're putting on a particular strain of bacteria for the particular crop you're trying to raise. So soybeans have a bit of bacterial inoculant that's different from other types of legumes. So you have to match up the crop you want to plant with the inoculant, make sure you got the right species. There have been a lot of fungal inoculants using mycorrhiza fungi as well, that are beneficial. The problem with the live culture approach is that you can either have competition from the native species, and there have been a number of studies that have documented that if you put out a bacterial or a fungal inoculant, where you've got a native population that's present in the soil, the native guys tend to drive off or compete with the ones that are introduced through the inoculant, and so they're not always terribly effective. In fact, there was a study reported in Corn and Soybean Digest over the summer about different trials done with bacterial inoculants, and they found that for most cases you don't get a yield response because of this competition issue. So very quickly the native guys can overwhelm any sort of inoculate species that come in. The other problem you have with live cultures is that they've got a relatively short shelf life, and they can't get too hot or too cold. And there's also evidence, in fact, one of the speakers at the National No-Till Conference this year talked about competition between fungicides that are applied to seed and live culture inoculants, that the fungicides actually might be deterring the performance of the inoculants, so that you actually should be getting a yield gain, and it might be setting you backwards because the two treatments are working against one another. So there are some issues there related to using live culture products. And then if we get to our area, which is the biostimulants, basically is when we talk about Chandler seed treatment products, we're combining growth hormones, enzymes, other biostimulants. Basically, it's a biochemical approach to try to multiply the beneficial microbes in the soil and promote plant health without having to put a live culture on it. And in many cases, what we found is that they're very reliable. The shelf life is very stable. The enzymes and the other biocomponents of these treatments can last for years. We've had people that had seed treatment for 4 years, did a side-by-side trial with fresh stuff, and had the same germination results, same rate of germination. So especially enzyme-based biostimulants for seed treatment can be relatively stable compared to the live culture approach. They're not as affected by having fungicides or other pesticides on the seed, since they're stable compounds by themselves. They're going to work without being overwhelmed. And there are a number of products in the market, including ours, that take this particular approach. So again, there are lots of products that fall under this biological seed treatment canopy. They all have different modes of action, and as Joel Gruber from Western Illinois stated at the very end of his presentation at the National No-Till Conference, is that not, I don't I don't want to borrow Joel's presentation. I certainly didn't get his permission in advance, but he provided very good advice to the crowd there, and said, if you're thinking about one of these products, the key thing to ask is how's it working, and what sort of mode of action does it have. I thought that was really good advice, that there are so many different products in the market, you have to figure out what it's really doing before you consider putting it on. And there may be interaction effects to consider as well. So there are lots of different treatments, and since they do work in different ways, you want to make sure that you're not going to do things that are working against one another, and because that's going to be wasteful of resources and money, and probably not the best for the soil biology and plant health, and ultimately for your yield. So I did want to take a little bit of time while we have a chance to talk about our particular seed treatment. We've been working with this since the late 70s on our own farm, and started marketing in 1981. We've had very good results over the years. Basically, it's a non-toxic enzyme-based seed treatment. It's available in either dry or liquid form, and it's applied directly to the seed. We'll talk about application here in a moment, but the three main groups of ingredients first are the biochemicals intended to support the soil biology, promote not only growth but reproduction of the microbes in the soil, the beneficial guys. So there are enzymes. There are also some amino acids and vitamin B complexes that help with the reproduction and growth of those colonies. We do include several chelated micronutrients, especially those that are critical for root and early plant development. So there's sulfur, zinc, boron, copper, which is very important for root growth, some iron, magnesium, manganese, and molybdenum. All of those are enzyme activators. They're present in very low concentration, much less than 1% content of the product. Um, if you have micronutrient deficiencies you're trying to fix in the soil, we're not going to provide enough to do that. These elements are only present in what's required just for germination and early plant growth. So it's not really a fertilizer. It's not going to amend any sort of shortage you've got in your nutrition, but it is going to make sure that there's enough of these micronutrients available to kick off those enzymes required for that early plant development. And then the last set of ingredients are the presence of cytokinins in particular, but there's also auxins and gibberellins present. So germination through the gibberellins, root growth through the auxins, and then early plant and shoot development through the cytokinins. All of those are present. There are a number of products on the market similar to ours. Our dry product is talc based. That's the carrier. People tell us, well, I've applied talc and I get a yield increase. Okay, well, that's probably because you're able to attract more moisture to that seed and start germination faster. So you get not only more uniform and more complete germination, but you're getting that plant out of the ground faster by simply having more moisture there. There are products that we've found that have just talc and the micronutrients, and those, by activating those enzymes and controlling the germination, probably are going to have a beneficial impact. But the key thing to look for on any label, and it's clear on ours, is the biochemicals, not only the plant hormones, but the enzymes, which help regulate not only the plant growth, but the microbial activity in the soil that supports it. So those three classes of ingredients are all really important, and especially the enzymes for early root development. What we've got here is a picture from one of our early side-by-side on-farm trials. The root system on the right is corn roots that are very deep, You can also see lots of lateral movement. Um, and the key thing is the large mass of root hairs. So the rhizosphere's got a lot of mass to it. There's a lot of surface area there for interaction between the microbes and the root system. The one on the right doesn't go nearly as deep. There isn't nearly as much root mass. Pretty good root system for corn, but it doesn't have the depth and the mass that's going to provide that interaction between the microbial populations, and as well sink down deep and be able to draw from subsoil moisture and nutrient sources if the season turns dry. So that's a fairly typical comparison that we see on side-by-side control versus treated for this particular product. A lot of times you don't see any difference above the ground. You have to dig the roots in order to see the difference. But then visually, we tend to see the differences at the end of the season when you make your yield comparisons. So it's not something that's visually happening above the ground because most of the action is taking place on the roots for this sort of a product. On corn, we typically get the plants up out of the ground 1 to 2 days faster because you've got those micronutrients available to help with enzyme activation, the hormones to help make sure the plants are growing, and as well the enzymes and the other things that help stimulate the microbes that are very much involved in root development. We typically, again, find not only deeper primary roots, but you get a wider spread of crown roots. That's getting to be a real issue, and especially corn farming, as producers are pushing higher and higher populations, there's less area between plants. People go into narrow rows from 30s to 15s and twin rows, all sorts of things to space those plants out more to provide more area for roots to develop. That's critical. It's it's a problem. We're not able to grow the root systems we used to because the populations are there. But nonetheless, on a side-by-side comparison, you should expect to see not only deeper root system, but a larger mass of hair roots, a much larger rhizosphere. Typically, we're going to get about 1 to 2,000 more plants per acre, so you've got more of those plants germinating and doing so more uniformly. We've had people that have had frost issues, and we've had observed up to 10 degrees frost protection on seedlings, mainly because you're getting more nutrient into the plant and producing higher levels of plant sugar that act as sort of a protection against low temperatures. Cool soils, warm temps, the plant's going to keep growing if it's got enough biological activity around the roots to keep feeding it, even though you're in difficult situations. On corn, normal years, we're typically going to see about a 6.5 bushel increase, and that's long-term average since the 80s. Dry years, or especially difficult years, will see much higher increases. 2 years ago, 2012, we had people growing corn that had almost normal yields. Not quite, but closer to normal yields than a lot of other folks that didn't have the root system and the biological activity to keep that plant going, even though it was very dry. We saw side-by-side yield increases up to about 50 bushels per acre. Very unusual, not the typical result. Somewhere in the 6 to 8 bushel range is much more typical. But if you get a dry year where the plant's extremely stressed, that additional root growth down into the subsoil allows you to seek additional moisture that's going to keep that plant going and producing yield when other plants are going to go dormant and start to die. So we've had very good results on corn. We've had also excellent results on soybeans. Again, faster emergence, less susceptible to extreme conditions. The deeper tap roots help with bringing water into the plant if things get dry. Typically get a boost in the germination rate, better pod fill, and more pods. Typically about a pound or two higher test weight. And the key thing is consistently anywhere from 20 to 40% more nodules. Usually it's around 30-35 is the average what we see when we count nodules on treated and untreated plants. And a lot of that has to do with the fact that you're sending out more flavonoids. That's one of the chemicals that the seed treatment helps to stimulate. And you've also got more rhizobacteria because you're promoting those populations. So you not only get stronger signals, but you get a better response. And so there's going to be more nodulation on the roots by promoting that biological activity in the soil with the treatment. In particular, we haven't done a whole lot of university research, but in the early years, one of the pieces that was done was to measure the different plant defense chemicals that are generated by bean plants after treatment, and then it was compared with plants that were untreated. The first one here, which I'm not sure I can even pronounce, is a diterpenoid, which again has antifungal and antibacterial properties. 55% increase over the control, 25% more tannins, which again deter insects. And the flavonoids have this good and bad character where they signal the good bacteria, call in the rhizobacteria. So a 40% increase is where we're getting the increased nodulation because you're better able to signal the good bacteria to come in and colonize that root system. And as well, the flavonoids deter the bad guys, especially pathogenic fungus. Um, More nodules are great, but what about nitrogen fixation? Well, in terms of the actual nitrogen fixation, after about the fourth week, when that colonization starts on the roots, it ramps up pretty quickly to somewhere in the 15 to 20% increase relative to the control. So you're producing about that much more nitrogen early. It does tail off to about 10 to 12, but a good season average would be about 15% more nitrogen fixed in treated legumes relative to plants that weren't treated with the seed treatment. So you not only get more nodules, but it pays off with more nitrogen. Wheat, rye, oats, other small grains, again, faster emergence. In this case, you get deeper root system with more tillers, better germination. You're going to have more uniform head formation and grain fill. Again, less susceptible to extreme conditions. Comparison, right is the treated, left is untreated. That's two areas, same area, but there's more plants, more tillers, and deeper roots where the wheat was treated. And this is one area where people growing cover crops have found really good success with using Chandler dry seed and liquid seed treatment. Especially if you're putting cover crops on late, not into ideal conditions, maybe you've got some other weather issues. As long as you can get those plants germinated and get the roots down, that's what you really want. You're not looking for that above ground growth so much as the root growth. And improving the germination on those cover crops is, especially given the cost, and if you're having to fly them on, it can get pretty expensive. You want to make sure you get as many of those germinated as possible. So not only in cash crops like winter wheat, oats, and rye, but also in cereal-based cover crops, we had really good results. In terms of the application rates and costs, really only need a very small amount. Our dry seed treatment is on a talc carrier, so you use 4 ounces per 50-pound unit. Some of the seed companies have gone to seed count units, but if you really just compare it in terms of a bag of corn or beans or wheat, you only need 4 ounces. That's where we start. For smaller seed like alfalfa, Austrian peas, other small seeded crops, you up that to 8 because you're dealing with more surface area. There's a lot of concern about talc and its interaction with the pesticides, the insecticides, and how that might affect beneficial insect species. For that reason, we tell people only put on as much as you need, just enough to cover the seed coat, no more. If you see any excess blowing behind the planter or the drill, or you're seeing an excess in the bottom of your planter hoppers, cut back. It does vary based on size of seed. It also varies a little bit with humidity of the day and things like that. We do have a liquid available. It's a little bit cheaper, but you have to have liquid treatment equipment. So unless your seed dealer is willing to put it on, some are, some aren't. Some people have their own liquid treatment equipment. If you have that, it's a convenient way to do it. Then you don't have the talc issue, and you do save a little bit of money. In terms of cost per acre on corn, we're talking about $1. If you go to the liquid side, it's going to be closer to about 75, 80 cents. Soybeans, depending on how heavily you seed, it's going to be in the $2 to $4 range. About the same for wheat, small grains, cereal cover crops, and then alfalfa, winter peas, any small seeded crop. Depending on how heavy you go, it's going to be about the $1 to $3 range. So the one comment we get from all of our customers is, hey, this is a no-brainer. For a very small investment per acre, I'm going to make sure that I got my soil biology working with me. And if there's a problem, weather stress, anything else, I'll help to overcome it. It's not going to make up for all of the problems you could face in the year, but it's going to help make up for some of them. And with those yield responses, typically 6 bushels on corn, 3 to 4 on beans, normal years, and then bigger increases in stressed years, is a very good return on your investment. We did have a few questions provided in advance. I was going to try to answer a few of those while we have time, but as we conclude, I just wanted to say that our best price of the year is over. Our December discount is 12% off the retail price, but for anybody who's registered for the webinar, we'd be happy to extend that December price to you for any purchases made through the end of March.
All right, real good. Thank you, Doug. It's some real good review of science behind this. Appreciate that. A lot of in-depth information. And like I said, we do want to take about 5 minutes here of time to answer a few questions. And thank you to those who submitted questions ahead of time. Doug, I'm just going to turn this back over to you. And I know there's a couple things you highlighted that you may not have been able to cover in your presentation, or that you might want to expound upon. So I'm just going to turn that mic back over to you and let you kind of go through some of the questions here that we've received.
Okay, Daryl. One of the folks asked about including zinc with biologicals on seed, and as we said, zinc is one of the key micronutrients required for enzyme activation. It's also used, got other purposes in the plant. The amount of zinc we include is really just there for those early enzyme activities in root development and early plant growth. If you need additional zinc, you're going to have to get it from other sources. And we do have customers that are including zinc either as a seed treatment or with their starter, and it's not going to cause a problem with the seed treatment. One of the questions had to do about root mass in wide versus narrow rows, and as we talked, that's one of the real challenges in high population farming. If you're going to stick with 30-inch rows and start to push those populations well over 30,000 in corn, it's going to provide less in-row space for that lateral root growth, and that's where some people have had some standability problems, and so a lot of people are looking at 20-inch, 15-inch rows, twin row technologies, trying to space those plants out so they have more room for roots to grow. And as well, people have gone to controlled traffic and some other approaches that reduce the impact of wheeled traffic on root growth. And there were some folks at the No-Till conference this year that had some subsoil photos of root growth and the impact of excessively heavy machinery just on the ability of the roots to grow into the center of the field if you've got compaction issues caused by machinery. So it's a real challenge, and the more space you can provide those plants, you're going to develop that root potential much more. Some folks asked about the shelf life on treated seed. Again, as long as our product doesn't get too hot, and by too hot, we mean over about 130 degrees. The protein chains and the enzymes and the hormones are going to be fine. The product can freeze and it'll be okay. So as long as it doesn't get hot or it doesn't get wet, it's going to be shelf stable for at least 4 years. The same product is used on all seeded crops, so it's not germplasm specific. You can use it on wheat, corn. We've had people use it this past year on sunflowers and sesame. Sesame was a new crop for us. I don't think we'd had anybody tried sesame before. They had very good results. And it can be applied with any other seed treatments, whether it's an inoculant or a fungicide or an insecticide. It's typically mixed in the dry treatment in the box. People have also put it in bags or bulk boxes before they go to the field. And you can do that a couple of weeks before planting because the product's pretty stable. It's not going to cause any trouble with the seed by pre-treating it. The liquid product can be mixed with fungicides and other liquid treatments if you're doing that yourself. And so we don't have any real issues with other treatments. Let's see.
Why don't we take about two more? And if you want, there's a couple questions that also got submitted here during the presentation. I'll let you take a look at those real quick, Doug, and see if you want to answer a couple of those.
Okay, one of the questions about compatibility with poncho or cruiser treatments. Again, we haven't had any problems. We've got lots of people that are using those insecticide, fungicide products as well as Chandler dry seed treatment and liquid, and they're getting along just fine. So ours is stable enough; it's not affected by the chemical controls, the chemicals that are applied by the seed companies, or used to be by the folks themselves, but most of them get it treated already from the seed dealer. Is it more effective on conventional till or no till? We haven't noticed a difference. We've had good results on all sorts of tillage. Central fill air planters blow the seed treatment off. No, that the talc adheres to the coat of the seed. Air planters, people actually cut back a little bit. So if you have a white planter, for example, a lot of our customers are using about 3 ounces per unit of seed because the planter helps distribute it. If you notice a lot of treatment blowing behind the planter, or you've got an excess in the central fill hopper, you can cut back a little bit more. But usually on air planters, you can go at a slightly lower rate, but the air system's not going to blow it off. The talc will adhere. At trade shows, my dad puts some on his finger and rubs it off on his shirt or his pants, and you'll notice it's still treated on the seed. And so if you take a few seeds, especially those that are treated, that are purple or colored, and sprinkle the talc on it and try to polish it off afterwards, you'll see that it adheres very quickly. The talc, because it's fine, it's a clay-like material, it binds very well. Let's see, third-party research, and we've got lots and lots of third-party research. Most of it's on-farm research done by the producers themselves. They're reporting the results to us, so it's not us picking and choosing between results we want, but the averages I cited for yield increases come to us from the producers. We're always doing research. In fact, this coming year, we're going to have a plot on a private research farm administered by a farmer who does this for other companies. And we're going to test combinations of our seed treatment, inoculant for soybeans, a rhizobacteria inoculant, and then the combination of the two. Lots of our customers get by without inoculation on their soybeans as long as they've had at least beans in the field for the last couple of years, and they say they don't have to inoculate. In principle, the enzymes should help the inoculant work better, so you get better performance from your inoculant. So we're going to do a side-by-side inoculant versus seed treat, and then the combination. The customers tell us that they feel their inoculant works better when they use our seed treatment, but that's their comments and it's anecdotal. We want to know for sure for ourselves, so that's one piece of our research program for this coming year that we're having a third party take care of for us. So we'll report the results in our fall newsletter. One more question.
Sure.
And let's see, is the soil deficient for micronutrients? Are there responses every year? If the soil is deficient in micronutrients, we provide enough for germination and early root growth, not enough for full season micronutrient needs. And we do tend to see a response every year. The response tends to be larger in those years when you've got stress. If you have sort of an ideal year, I can't remember the last one we had, but in those years where weather works right, timing's right, you had not too many issues with crop production, you're probably going to see a smaller response because there are fewer challenges to respond to. When you get into stress, that's really when you see this sort of product payoff.
All right, real good. Thank you, Doug. There may be a few more questions here we're going to send home with Doug, and when he has a chance, we'll have him answer those, and we'll make those available through the webinar archive that we will be making available. Before we conclude, just real heads up that when you log off today, there's going to be a questionnaire we're going to ask you if you would complete. It's a very simple, easy to complete, small number of questions. And those answers help us determine the value of today's webinar and also help us with planning for future ones. Second, as I mentioned, we're going to make available via email to you a replay of today's webinar in the event you'd like to go back and review the entire presentation or parts of the presentation. You'll also find that replay of the webinar up on our No-Till Farmer website shortly. There's an archive section on the website. You can go back and listen to other webinars that were recently completed. And as I mentioned, we're going to send any of these other questions back with Doug and get those posted as soon as we can. So I do want to thank Doug for leading this discussion and also thank Midwest Biotech for sponsoring this webinar on biological seed treatments. I also want to recognize and thank today's producer, Cole Vandermouse, for steering us through the webinar today. And thank you for joining us. And hey, have a good rest of winter. Hope it's short for you. And I know you're all looking forward to the 2014 production season. Take care and hope to see you on another webinar soon. Bye-bye.